Good morning, everyone, wherever you may be in the country or overseas. I hope you're well. I am Caleb Don Conyate. Welcome to the third session of the 20th Annual Symposium of the Philippine Network of Microbial Culture Collections. This year's month-long event is centered on the theme 3Cs, Culture Collections and COVID-19. This session's topics are at the heart of the pandemic that is crippling the globe as of the moment. The two topics are COVID-19 vaccines, where are we now and what is next? The other one is vaccine platforms for COVID-19, which ones to choose and why. We will now officially start the program with the invocation to be led by Mr. Ian Lana of the TUA Christian Education Department, after which the Philippine National Anthem will be rendered by the Trinity University of Asia Chorale.
A pleasant morning to all of you. It is good that we start our activity this morning by recognizing the Lord in thanksgiving and prayer. The Lord has given us a new day, and so it is right to thank Him and to rejoice and be glad in His goodness. To start with, let us sing together as we prepare our hearts and mind in prayer.
I am the God that healeth thee. I am the Lord your healer. I sent my word and healed your disease. I am the Lord your healer.
As we come before the Lord in prayer, allow me to read a passage in the Scripture and let the Word of God encourage us this morning. Praise the Lord, my soul, all my inmost being, praise His holy name. Praise the Lord, my soul, and forget not all His benefits, who forgives all your sins and heals all your diseases, who redeems your life from the pit and crowns you with love and compassion, who satisfies your desires with good things, so that your youth is renewed like the eagles. Heal me, Lord, and I will be healed. Save me, and I will be saved, for you are the one I praise. I will restore you to health and heal your wounds, declares the Lord. Come, let us return to the Lord. He has torn us to pieces, but He will heal us. He has injured us, but He will bind up our wounds. Let us pray. Our Heavenly Father, Creator of everything visible and invisible, who made the vast universe and microscopic world, we come to you in humble recognition of who you are, our great and awesome God, holy and almighty. Thank you for the wonders of life and the beauty of your creation. We praise and worship you, for you are good, you are merciful, and you are faithful in all your dealings with us. Now, Father Almighty, as we continue with our symposium with the theme 3Cs, Culture, Collection, and COVID-19, and as we discuss also the development of a vaccine for COVID-19, we pray and humbly ask your blessing in our activity today, that it will be a source of great discovery, enlightenment, hope, and encouragement to all who are here today. All wisdom and knowledge come from you alone, and so we seek your favor, that in due time you will answer our prayer to bless the collaborations of doctors and experts around the world to create a vaccine that will stop spread and protect us from this virus. We now ask, Father in heaven, through the inspiration of the Holy Spirit, to make this symposium successful and productive for all of us who are here today. Guide and empower our speakers today. Give them your wisdom to communicate the truth in their field of expertise. We lift to you our audience as they engage in the discussions. Open our minds to new ideas and discoveries and give us a new perspective to understand the nature of your creation in the world of microorganisms. We lift to you the organizers of this event. Would you bless them for their hard work and dedication? And now, as we start our program, we pray for the unity of minds and hearts, for the glory of your name. Bless us, Lord God of heaven, with joy that comes from your Holy Spirit, and with the peace of the Lord that transcends all human understanding. And all this we pray in the name of our Lord Jesus Christ, who lives and reigns with you together with the Holy Spirit, one God, forever and ever. Amen.
Bayang magiliw, pero sa silanganan, alam ng puso, sa dibdib mo'y buhay. Lupang hinirang, tuloy ka ng magiting, sa manlulupig, di ka pasisihin. Sa dagat at bundok, sa simoy at sa langit mo buhaw, may dilag kang tula, tawid sa paglayang minamahal. Ang isang ng watawat mo'y tagumpay na nagdidinig. Ang bituin at araw na kailang pamay di magdidilim. Lumay ng araw na walang ating pagsinta. Buhay langit sa piling mo. Ang ligaya na pag may mga api, ang mamatay ng dahil sa'yo.
Thank you very much, Professor Lana. Thank you very much, TUA Corral. To formally welcome everyone to today's gathering, here is the chairperson of the Department of Biology of Trinity University of Asia, Dr. Anacleto Argayosa.
So good morning. Welcome to today's webinar series of the Philippine Network of Microbial Culture Collections Annual Symposium in collaboration with the Trinity University of Asia College of Arts, Sciences, and Education in the Biology Department. With the active support of the President, Dr. Wilfred Chu, the VPAA, Dr. Gisela D.A. Luna, and Dean Howell Ho. This is the third of the series of the PNMCC webinars, and today's topics on the development of vaccines for COVID-19 are very relevant and interesting. It is a great privilege to co-host a series of talks and has contributed to the understanding of the scientific community on many aspects of microbiology. So once again, on behalf of the TUA, we welcome our distinguished guest speakers and all the participants who are with us this morning via Zoom and FB live streaming for the 20th PNMCC Annual Symposium. Thank you.
Thank you very much, Dr. Argayosa, the PNMCC president and member of the faculty of the College of Allied Medical Professions of Angeles University Foundation, will now deliver the opening remarks. Please welcome Professor Diane Lagason Dizon.
Good morning to everyone. On behalf of the Board of Directors of the Philippine Network of Microbial Culture Collections Incorporated, it is my privilege and pleasure to welcome all our distinguished guests to the 20th PNMCC Annual Symposium Webinar Series. PNMCC is committed to lead in the standardization and upgrading of procedures for the isolation, characterization, conservation, and distribution of microorganisms and biosafety through trainings and seminars. Our task is to provide awareness and gives emphasis on the importance of microbial culture collections and safekeeping of such, especially during this time of pandemic. As the network marks its 20th symposium anniversary, PNMCC chose the theme 3Cs, which stands for Culture Collections and COVID-19. This year's annual symposium will be different from the past symposiums because it will be held virtually and it will be comprising of webinar series which will be held on the 4 Saturdays of October. The webinar series will address issues and concerns regarding the very controversial COVID-19, giving highlight on topics related to the testing of virus, updates on the development of vaccines, the role and importance of biosafety and biosecurity in the laboratories, and the urgency for the establishment of the Virology, Science and Technology Institute of the Philippines. We are now on the third day of our webinar series, and we had invited prestigious speakers who are going to share their knowledge and expertise, giving emphasis on the need for the development of COVID-19 vaccines to combat the serious disturbance of human activities and the increasing cases of fatalities. Another important topic that will be tackled this morning will give importance on which vaccine platform should be used for the development of COVID-19 vaccine and their importance. Also, the significance of the network of cells and microbial culture collections on vaccine development will be highlighted. Truly, these topics are very important and timely in this pandemic crisis we are facing too. I sincerely thank our honorable speakers for sharing their invaluable time and effort with us. I also want to thank our valuable sponsors for their generous contribution and support to the network by subsidizing financial assistance for this symposium. To the administration, faculty, and staff of the Trinity University of Asia, College of Arts, Science and Education, Biology Department, thank you very much for collaborating with us by granting our requests to use their platform, Zoom platform, and social media accounts for the promotion and during the actual presentations of the webinar series. We look forward to more collaborative work in the coming years. And to the most dedicated and hardworking Board of Directors of the PNMCC, my sincerest gratitude for making this event a success. With that, I welcome again our dear participants and guests. May you gain comprehensive knowledge with a feeling of satisfaction for participating in this webinar series. Thank you and a pleasant morning to all.
Thank you very much, Professor Dizon. Before we begin, may I just remind everyone on the webinar guidelines. Please make sure that we carry ourselves in a respectable manner throughout the event. Everyone is encouraged to speak in English. Please turn off your webcams and put to mute your microphones while speakers are presenting. This will lower the bandwidth and prevent background noise. Please send questions and comments through the chat box, which are screened by our facilitators. Please know that due to time constraints, only a few questions will be entertained later. Please allow me to thank our chat moderators, Professor Anthony Alcantara for Zoom, Professor Hazel Yawe Arce for Facebook, and they will be joined by Professor Nick Sean Tabao and Mr. Carlo Chris Aporilio. As we are now very ready and deeply excited to listen to the speakers, we also have our questions on the topics that will be addressed. We will now give the time to the webinar moderator and our esteemed speakers. Please welcome our webinar moderator, University of the Philippines, Manila professor and university scientist, and also the former PNMCC president, Dr. Marilyn Parungao Balolong.
Morning. Thank you for that, Caleb, for that introduction. So I will be introducing our first speaker for this morning. So Dr. Siddharth Shidar is a clinical assistant professor at the Department of Microbiology of the University of Hong Kong. He is a clinical virologist with research expertise in emerging viral infections and laboratory diagnostics. Dr. Shidar has been involved in several studies examining animal models, seroprevalence, molecular diagnostics, epidemiology, infection control, and clinical manifestation of COVID-19. He has used his full picture view of the disease to engage the public on controversial aspects of COVID-19 via the talks and social media. So everyone, please join me in welcoming our first speaker for this morning, Dr. Siddharth Shidar, for his talk entitled COVID-19 Vaccines, Where Are We Now and What's Next? A warm virtual welcome or physical applause, please, for Dr. Shidar. Dr. Shidar, you may take the stage.
Thank you very much indeed for the kind introduction as well as the invitation to this wonderfully organized webinar. Let me just share my screen now. Right, I hope everyone can see the shared screen, and I will begin. So my topic today is indeed on a very cutting edge, controversial side of the pandemic that we are currently facing. It's on COVID-19 vaccines, where we are now and what's coming up next. So before I begin, let me just declare conflicts of interest that I may have. I've received speakers honoraria from Abbott Medical Laboratories and Sanofi Pasteur within the last 5 years. Right, so I'm sure I don't need to talk about this too much because we've been hearing about nothing else but coronaviruses for the last 9 months or so. But a quick introduction to this wonderful family of viruses. It's actually a very large group of viruses that cause infection in many, many animals, including humans. Before COVID-19, SARS coronavirus 2, we had 6 coronaviruses that were known to infect humans. The coronavirus structure, as you can see here, is very, very small, being a virus. In the range of about 100, 120 nanometers in terms of its width. And you can see these little jutting out bits from the coronavirus. And this is called the spike, right? You can see it here and more nicely in the schematic diagram here, you can see the coronavirus spike protein. The spike protein is particularly important because the spike protein engages with the receptors on human cells. And not only that, it is the most important target for our patients' immune response against the virus. So I'll be talking a lot about the spike protein. And when I say spike, this is basically what I mean. In late 2019, something unusual happened, as we all know. In China, they started noticing a lot of people having pneumonia of unknown cause, unknown etiology, and they found that a lot of those cases had actually recently visited the seafood wholesale market in Wuhan. And this eventually led to the discovery of the new coronavirus, which is SARS coronavirus 2. SARS coronavirus 2 has proven to be highly adapted to humans, and it is readily transmissible between humans. Just like SARS coronavirus in 2002 to 2004, the SARS coronavirus 2 uses the same cell receptor, which is called ACE2. By this, I mean that the spike protein of the SARS coronavirus 2 engages with the ACE2 receptor on the surface of cells. We are still unsure where SARS coronavirus 2 comes from. We are pretty certain that the ultimate ancestor is a bat, given that the virus is very close to other coronaviruses detected in bats, but we don't know whether there was some animal involved in the middle, and some people have referred to pangolins as a potential intermediate host, but we're not sure as yet. But since its emergence, it has caused a lot of devastation around the world, extreme disruption, and as of this last week, there have been in excess of 37 million cases and more than 1 million deaths. And as we speak, most regions in the world are actually struggling with control of COVID-19. Now, what are the strategies for COVID-19 elimination or control that we have so far? The one that we're all familiar with are the public health interventions. Public health interventions include a variety of things like closing down borders or controlling the flow of people between regions. The second step is testing and tracing. So you detect as many cases as possible. And once you detect cases, you isolate cases and quarantine contacts of those cases. So this is bread and butter of epidemiology and has proven really valuable in controlling COVID-19, especially in this region, in East Asia. Social distancing measures we're all familiar with. They include various things like school closures, closures of various public facilities, other indoor areas, etc. Widespread use of face masks and increased hand hygiene by members of the general public is something that we're seeing very commonly. And a lot of enhanced infection control measures in various healthcare settings. And last but not least, we have lockdowns, right? So this is an extreme measure that is used to clamp down on community transmission, and it's something that we don't like to see very much. But many parts of the world are forced to lock down on an intermittent basis to control COVID-19 spread. But these are short-term strategies. What strategies do we have to control COVID-19 in the long term, right? One of them would be a development of effective medicines or antivirals, but I must say that that has been extremely disappointing and difficult to date. So just yesterday, you would have heard that one of the main candidates called remdesivir was actually not shown to have any benefit in terms of preventing death for COVID-19. So that was a big disappointment to many, many people. The eventual ticket out of the COVID-19 pandemic is touted to be vaccines, right? So we believe that if we produce an effective vaccine against COVID-19, that would be an extremely useful tool to cut down COVID-19 transmission. And theoretically, that makes sense because vaccines have helped us with so many diseases in the past, from smallpox to polio, etc., right? So this talk is going to be focusing on vaccines for COVID-19, what we know about them so far, and what I think is going to come up next. Before I begin talking about vaccines, I just want to say that the pace of development of vaccines has been breathtaking, right? We have had more than 100 candidates, easily more than 100 candidates of vaccines being developed by various universities, companies, institutions all around the world. This has been a tremendous amount of investment at a very fast pace, and it's wonderful to see in a way. A very useful resource is the New York Times Coronavirus Vaccine Tracker. And here you can see how many vaccines have entered different phases of clinical development. And you can see that as of last week, we already have 11 candidates in very advanced human trials. So that's great to know, right? And a few of them have also received what we call limited registration, which means that they can be deployed in certain settings, like for healthcare workers or for army personnel, etc. And a few countries have decided to expedite some limited registration for this. But as of now, there are no coronavirus vaccines, COVID-19 vaccines on the market. Now, I just like to start off on a slightly negative note, but I think it's important that I clarify these things before starting to talk about individual vaccines. So the title of the slide is, how can vaccines help in combating COVID-19, or can they help in combating COVID-19? There is a general acceptance among the people developing these vaccines that COVID-19 vaccines would not be 100% effective. This is a very fair thing to say. Almost no therapeutic intervention is 100% effective. There's no 100% guarantee for anything in this world, right? But what they've done is they've leveraged the fact that we have never developed a coronavirus vaccine in human history, right? We don't have super effective vaccines against any respiratory virus. Think about the influenza vaccine. The overall efficacy of the influenza vaccine is about 50% there and thereabouts, right? So based on this, the WHO has said that you can say that you have a successful COVID-19 if there is risk reduction by 50%. So what that means is if somebody gets the vaccine and they're 50% less likely to catch symptomatic COVID-19, then you can say you have a successful vaccine. That seems to be a pretty low bar, but it is actually quite a realistic one. And as I show in coming slides, you'll realize that that is actually a very wise way of determining an efficacy cutoff, right? Now, when I say efficacy, we also have to appreciate that there are many different types of efficacy. Everybody talks about effective vaccines or efficacy, but what do we actually mean when we say that? Generally speaking, when we talk about efficacy, we're talking about the second point here. It means that does the vaccine prevent symptomatic infection in people who have received the vaccine? That is, if I get the vaccine and if I'm exposed to COVID-19, would I get a fever, would I get a cough, or would I be completely asymptomatic? That's what symptomatic infection is referring to. But remember that there are more stringent definitions of efficacy, which is sterilizing immunity. The best vaccines produce sterilizing immunity. What that means is once you get the vaccine, if you're exposed to the virus, you're not just protected from symptoms, you're protected from infection itself. You're not going to get infected at all. The virus simply cannot enter your body. That is what we mean by sterilizing immunity. Many vaccines, such as say the hepatitis B vaccine or the hepatitis B vaccine is a good example of that. They offer very powerful immunity, almost sterilizing immunity. There's no question of being infected by the virus itself. But it remains to be seen whether the same holds true for COVID-19 vaccines, okay? So this is a very high bar, the sterilizing immunity. Other measures of efficacy would include prevention of severe infection. So, okay, they may not protect you from getting infected, but would they protect you from catching severe infection? That's useful, isn't it? So if you catch COVID-19, but if you're protected from severe infection, then most people would still choose to get the vaccine. Hey, it's just a cold-like thing, right? So it makes sense to get the vaccine to protect yourself from severe infection. However, I'd like to point out something, that unless the vaccines are able to give you sterilizing immunity, it is going to be difficult to control the pandemic. Because if people get the vaccine and they still catch the infection, then theoretically they're still capable of transmitting it to others. So when we talk about COVID-19 vaccines as a way of controlling the pandemic, we have to be realistic about what we are expecting from the vaccines, if you catch my meaning. So if the vaccine offers less than sterilizing immunity, COVID-19 vaccines may not be able to bring down community transmission to zero, right? So we're just keeping things realistic. So other measures of efficacy would include your so-called indirect protection, which means that if I get the vaccine, would my family be protected to some extent against COVID-19, right? So it's a kind of herd immunity. How effective are COVID-19 vaccines at getting herd immunity. And also vaccine efficacy in the real world also needs to be studied. Now, I'll say one thing, that most trials, clinical trials on vaccines to date, are looking at the prevention of symptomatic infection as their primary outcome. They're mostly interested in whether or not people who catch COVID-19 after getting vaccinated have symptoms or not. Some vaccine trials would also be looking at severe infection. In fact, most would be looking at severe infection as a secondary outcome. And some may also have ways to see whether or not their vaccines achieve sterilizing immunity. That is by seeing whether or not people actually catch the infection at any stage after vaccination. So we're going to get some of this information soon, right? But their contributions to herd immunity or real world effectiveness is going to take much longer to actually determine. Now, how do we test vaccines? Well, there are many stages. Traditionally speaking, these are the steps. The first step is a preclinical animal study, which means that we give the jab to animals and then we challenge them with a strain of the virus in question and we see how effective the vaccine is. Then we progress to what are called phase 1 or phase 2 trials, that are trials to basically determine the optimal dosage, get an very, very approximate safety profile, and get a few immunological correlates of protection, by which I mean how well do people respond to the vaccine in terms of antibody production. These are things that are looked at in phase 1, 2 studies. Phase 3 are what we call our randomized controlled trials, right? So these are actually trying to determine efficacy, okay? So the outcome of a phase 3 study is to look at vaccine efficacy. Phase 4 is what happens after the vaccine is marketed. People look at how effective it is in the real world, how safe it is in the real world. So it's a very long-term thing. Now, in terms of how we measure vaccine efficacy, as I told you, in phase 1 and phase 2, we're mostly looking at antibody responses. We're also looking at something called T-cell responses. So if you know, the immune response has actually two arms. It has the adaptive immune response has a humoral immunity arm, which is antibodies, and a cell-mediated immune arm, which is mediated by immune cells called T cells. So these are what we look at in phase 1 and phase 2. As I mentioned, vaccine efficacy is something we're going to find out in by randomized controlled trials, and safety signals are something that are constantly evaluated. So the two items that I mark in bold, the antibody responses and T-cell responses, are things that we actually already have a very good view about. Okay, now just to again reiterate the point that the pace of vaccine development has been very breathtaking and exciting, right? The traditional paradigm of vaccine development honestly, it takes a decade or so. You identify your targets, you go through all the preclinical, clinical trials, you set up manufacturing, scaling up, validation of process, large-scale manufacturing, licensing. Anyway, the whole thing takes ages, full of bureaucracy, and actually most vaccines die before they ever come to market. It's that difficult, right? But for the COVID-19 pandemic, we really learned our lessons and everything is being streamlined. Things are happening at the same time. The preclinical studies, we already know because we've actually used these vaccine platforms in animal studies before, so we know how they work. In terms of the human trials, we are combining phase 1 and phase 2 together. We are constantly evaluating safety signals. At the same time, companies are talking to governments about authorization. We're talking about developing manufacturing capabilities even before vaccine efficacy data is available. So I can tell you there's a lot of money being gambled on vaccines that may or may not be effective at the end of the day, because people realize that if this vaccine turns out to be effective, then you need to get it out there as soon as possible. So I mean, the scaling up, manufacturing development, all that is being actually pushed way back, much more temporally, much more back than it usually is, right? So it's something that is honestly a credit to the pharmaceutical world in general as to how quickly they have responded to this. Okay, now in terms of vaccine platforms, let me just get rid of this here. In terms of vaccine platforms, one thing that I would not be doing during this talk, at least, is to compare different platforms, right? I think in the following talk by Dr. Gloriani, she'll tell you a lot about how these platforms kind of match up against each other. But I personally would just focus on 4 types of vaccines. There are many different types of vaccine designs out there for COVID-19, but I'm just going to focus on 4 types today. The reason I focus on these 4 types is not because I think they're going to work, is because these are the front runners in a way in terms of having the most data out there, right? So many of these vaccines are still in preclinical or phase 1. We simply don't know much about these vaccines. So I would not be referring to those, but I'll be referring to those that I personally know to be in phase 3 already. So these are vaccines for which are promising, okay? So the 4 types are inactivated vaccines, vector vaccines, spike-based vaccines, and mRNA vaccines. So I'll go over them one by one, along with a brief summary of what we know about the performance of these vaccines, right? Okay, so the first type are inactivated vaccines. These are beautifully simple vaccines, right? What we do is we get the SARS coronavirus 2 from an infected human. We grow them in a cell line, and then we characterize these viruses thoroughly. We know all about their genes, we know everything about them, right? And this, again, I want to highlight that this is the importance of culture collection. So you really, really need well-characterized virus strains, without which this kind of vaccine would be impossible. Then what we do is we so-called kill the vaccine or inactivate the vaccine. Very simple process with something like BPL, beta-propiolactone, which has a decades-long history in vaccine development. And then we combine it with an adjuvant, something like alum, again, a very, very common vaccine adjuvant. And that's it, you have your vaccine, right? And then you just jab it into a person. So the person is being exposed to the dead version of the vaccine rather than the live one. So that's the difference, right? 2 candidates from this part of the world are Sinovac's CoronaVac, and Sinopharm. So both of these are in phase 3 clinical trials. Right. Now, what do we know about how well these vaccines work? I think very useful information comes from the monkey studies. Okay, we don't actually have phase 3 trial data for either of these vaccines or any vaccine to date, but we do know about how they work in monkeys. So both vaccines, both Sinopharm as well as CoronaVac, have been tried out in monkeys. What we know is after receiving the jab, the monkeys do produce a good antibody response. And after being challenged with a virus following vaccination, the vaccinated monkeys respond better in terms of having less severe disease. So it does seem to be protective against severe disease. So you can see a placebo monkey gets very severe disease, a vaccinated monkey, the lung is less damaged after infection. But if you look at this particular graph, this compares the viral loads of SARS coronavirus 2 in the upper respiratory tract in the throat of vaccinated monkeys. Sorry, you can't see the panel here, but the red line and the blue line are the vaccinated monkeys. The green lines are the placebo monkeys. Now you can see that the viral load is actually higher in the placebo monkeys compared to the vaccinated monkeys. Good. However, what you can see from this is that vaccinated monkeys also do get infected, right? So this kind of protection is protection against severe disease, protection against symptomatic infection, but it does not seem to be sterilizing, in that vaccinated monkeys still get infected. It's just that the disease is less severe. So going back to the point I was making earlier, how well is this going to contribute to herd immunity is a question that needs to be answered, because you're protected from symptomatic infection, but you're not necessarily protected from infection, okay? And if you're infected, there is a possibility that you can still transmit to others. But you might argue that if the viral load is lower, then you're less likely to transmit it to others. But we're not sure about this. So this is something that is a repeated pattern in COVID-19 vaccine studies, as I'll illustrate. This is what we call a phase 1, phase 2 kind of study. This is the data for Sinopharm. I don't present the data for CoronaVac because it's actually very similar. Following the kind of vaccine regimen of 0 and 21 days apart, subjects do tend to get an okay kind of antibody response against the vaccine. But whether this actually protects against infection, we're waiting for phase 3 trials to actually determine. Okay, the second vaccine type are something called vector vaccines. Now, these are very exciting kind of cutting edge vaccine designs. There are no human vaccines based on this technology ever licensed, okay? But basically what they do is they use another virus. This is called an adenovirus. In the case of most COVID-19 vaccines are using an adenovirus vector. What the adenovirus vector does is that it carries a little bit of the SARS coronavirus 2 gene called the spike gene. So it's the gene that encodes for the spike protein. So it's delivered into the body. It traffics the spike gene into the cells. And then the spike gene is expressed to form spike protein, and that elicits an immune response. There are a lot of vaccines currently in phase 3 using this technology. The most famous, I guess all of us have heard about the AstraZeneca University of Oxford vaccine, and also the CanSino product from China, the Gamaleya product from Russia, and we have the Johnson & Johnson vaccine candidate as well. So again, I look at the monkey data, right? So these are vaccinated monkeys. They get a good antibody response. There's no doubt about it. But we're interested in what happens after they're infected. Now, after they're infected, there's no doubt that they get less severe disease. If you look at the lung fluid, so this is fluid right from the lungs, the viral load in the lungs is much lower than for the placebo monkeys. So it seems to protect them against a lot of viral replication in the lower respiratory tract. But what happens in the nasal swabs? Okay, so these are swabs from the nose like people get when they're getting tested for COVID-19. Again, you can see that a lot of the vaccinated monkeys, either receiving at a single dose or 2 doses, actually do have a little bit of virus replication going on in the nose. Again, the same point. It's protecting them from severe disease and probably symptomatic infection. We don't know because they're monkeys. But is it actually protecting them from infection and transmission is another question. This is the Johnson & Johnson product. Again, nasal swab, you get 1 monkey showing a little bit of infection and replication. So this is something people are a little bit concerned about. The phase 1, phase 2 data, this is for the Oxford candidate. I would say that it's looks okay. You get a good antibody response, and they also looked at T-cell responses, which were pretty robust. And it's very comparable to people who have actually recovered from COVID-19. So looks good, but how effective it's going to be, we need phase 3 to determine. Now, I must point out that not all adenovirus vectors are the same. The Oxford candidate used something called a chimpanzee adenovirus. So it's using an adenovirus from another animal. Some companies like the CanSino Biologics from China are using adenovirus 5. Adenovirus 5 is a human virus. Now, when they were recruiting candidates, they did something very interesting. They looked at how many people actually have antibodies against the vector. So it's a thing that's actually carrying the spike gene into the body. Theoretically, if you have an antibody against the vector, then the vaccine is going to be less effective, right? Because you're actually neutralizing the vector before it can enter the cell and deliver the gene to cells. So that's kind of bad news. Unfortunately, what they found that, yes, that is the case. If you have antibodies against the adenovirus type 5 vector, then you're less likely to respond to the vaccine. And this is particularly true in older people who most often have been exposed to adenovirus 5 in Asia, right? But they're still proceeding to phase 3 trials to see if this actually makes a real world difference in terms of vaccine efficacy. But that is something that we have to bear in mind. Not all vectors are the same. The next one are the mRNA vaccines. So mRNA vaccines, and what we do is, this is a beautiful example of nanotechnology. So we're using little snippets of the viral gene that encodes a spike protein. We actually put it in little things called lipid nanoparticles. The mRNA itself is very cleverly modified so that it can enter cells unimpeded, and it can be translated to form spike protein, and it can then trigger a very robust immune response. But I must point out that although this is very exciting, there are no licensed human vaccines using this technology. And the kind of disadvantage of mRNA vaccines is that you need a very stable cold chain, by which I mean you need freezers to keep these vaccines fresh. And you can imagine if you're trying to deliver a COVID-19 vaccine to a village somewhere, or somewhere that is 2 or 3 hours away from any major town or city, is going to be difficult. It's going to be very difficult to make sure that it remains frozen and stable all the way through. So that's a little bit of a problem. 2 major candidates, very famous as well, the Moderna from the US and the BioNTech Pfizer product. I'm not going to repeat myself here. It's actually the monkey data again. Same story. You get good antibody responses, you get good T cell responses, but it's not exactly sterilizing, okay? So you still get a little bit of virus replication. Again, how meaningful this is, we don't know, but it's just something that we take note of. This phase 1, phase 2 data for both of these vaccines have already been published. A thing about the mRNA vaccines is that you tend to get a lot of side effects. The vector vaccines as well. Inactivated vaccines are really, really comfortable to get because you don't have a lot of local symptoms. But the vector vaccines and the mRNA vaccines, you can expect to get a bit of local pain. You can expect to get a bit of fever and chills, quite stimulatory to your system. That may be a good thing. It may be that the more symptoms you have locally or systemically, it means that the vaccine is working better. But we're not sure about this, and hopefully we'll find out soon. The last type of vaccine that I'd like to talk about is the subunit vaccine. Basically what this is a modified recombinant spike protein. So basically we just cleverly modify the spike protein of the SARS coronavirus 2, and we adjuvant it with something called Matrix M1, which is a company-specific adjuvant by Novavax, and is then given to people as an intramuscular injection. Why I bring up this particular product is that to date, this is the only vaccine candidate that seems to show sterilizing immunities in monkeys. So that means monkeys that get this vaccine do not seem to get infected as far as nasal swab is concerned. So this would have an effect on these monkeys being able to transmit the virus. Okay, so that looks very interesting. But you'll see from here in the lung fluid, surprisingly, one monkey did have a little bit of replication. So we don't know, right? But it's something that is very interesting and noteworthy. The phase 1-2 data also looks very good. Now, I don't directly compare antibody responses between papers because it's impossible to compare, because different papers are using different methodologies. And unless you have a head-to-head comparison done in the same lab, you can't compare whether one vaccine is producing more antibodies than the other. So all I can say is, looks okay. I mean, they definitely do get antibodies after about 4 to 6 weeks of vaccination, no doubt about it. Now, all along I've been talking about jabs, okay? So these are vaccines that are given as injections, as intramuscular injections. Now, one thing we understand about respiratory viruses in general is that in the upper respiratory tract, you need something called secretory IgA. You need something called secretory IgA. So these are antibodies that can neutralize the virus in the upper respiratory tract. We know that jabs actually are very good at generating a different kind of antibody called IgG, right? That actually protects the lower respiratory tract. So this kind of explains the monkey data. It might be that the way we are giving the vaccines as intramuscular vaccines is actually protecting the lungs, but it may not be as good at protecting the upper respiratory tract. And so you get a little bit of infection and replication going on in the upper respiratory tract. So a logical way to get past this would be to give the vaccine as a spray, nasal spray. So many of you may be aware that we do this for the influenza vaccine. We have a nasal spray that can actually generate a fairly good immune response. So people are actually trying the same for COVID-19. This is also a chimpanzee adenovirus vaccine, not the same as the Oxford candidate, that is actually given into the nose instead of as a jab, right? And they found that actually it was quite protective against both upper as well as lower respiratory tract. So at the end of the day, we might find that the second generation of COVID-19 vaccines may not be jabs, but instead they may be nasal preparations. And physiologically, that makes a lot of sense. Right, so I'm getting towards the end here. So what do we know so far? We know that there are a variety of vaccine designs. In monkeys, they're able to reduce severity of disease. They're able to reduce but not eliminate infection and viral load shedding. When given to humans, these vaccine designs incite good short-term humoral and T-cell immune responses. In the short term, the vaccines do seem safe. Some produce more symptoms than others. What are we going to find out? By the end of November, we should be able to have access to the first phase 3 efficacy and safety data, and we should be able to answer the question whether some vaccine types are better than others. So this is really like a race, right? And it's really exciting to see which vaccine candidates are going to perform better than other. Over time, maybe in 2021, 2022, we're going to know about the type and durability of protection. But one item of concern is the details on distribution of vaccines, how they're going to be distributed, who is going to get it first, tremendous uncertainty. The WHO just came out a couple of days back saying that it's possible that most people would not have access to the vaccine until 2022. And we may be targeting specific population groups for vaccination first. So again, a lot of uncertainty with regards to that as well. How the vaccine is going to impact on COVID-19 in terms of COVID-19 pandemic is again something that depends on a lot of things that I hope the audience is able to appreciate. It depends on how effective these vaccines are at preventing infection and transmission. So it's something that remains to be seen. I believe that despite this, vaccines are definitely going to be a tool to adapt to the post-COVID era. I don't believe that COVID-19 is going to go away ever. It's our best hope is that it becomes one of the usual seasonal respiratory viruses that we all catch once in a while. Hopefully the severity is reduced by vaccination. That seems to be the trajectory of where we are heading. I would like to end by re-emphasizing that none of this work, this excellent cutting-edge scientific work would be possible without culture collections. You need standardized SARS coronavirus 2 strains for everything from vaccine preparation to testing the vaccines to testing antibody responses to the vaccines. In addition to the vector vaccines, you need well-characterized vectors, and cell culture collections are equally invaluable in performing things like growing the viruses or in performing things like neutralization testing. You can't do it without cells like the good old Vero, right? Which is the cell line that is recognized by the WHO for producing inactivated vaccines. Okay, I think I've overrun by a couple of minutes, but that is all I have to say today. And I'd be more than happy to take any questions from you either today or on other platforms. So I put up my email and Twitter handle up here. So feel free to approach me at any time with your thoughts and questions. Thank you very much.
Thank you very much, Dr. Sidar. That was a very interesting talk. I'm sure many of us are have excitedly prepared their questions. No? So I have here some set of questions. So let me start first. And for everyone who would still like their questions to be thrown out to Dr. Shidar, you can, I think, put them in the chat box, and then the moderators will be getting them and feed them to me. Okay. So, Dr. Shidar, for your first question, how long can herd immunity take effect once rolled out using these new generation vaccine designs? This is as compared to the traditional vaccines of long time ago. Or is this even, or is herd immunity even possible for COVID-19?
Yeah, that's a great question and a big unknown. My guess is that we're eventually going to achieve herd immunity against COVID-19. This is going to be by a combination of vaccination and probably some degree of natural infection. Eventually, enough people catch COVID-19, or eventually enough people are vaccinated to reduce the viral load of shedding if they get infected by COVID-19. And once this reaches about 50 to 60% of the population, you're going to see herd immunity. And on the trajectory that we are, if you add in a reasonably effective vaccine, that could happen fairly quickly. So that would be my best guess. So hopefully by next year end or the beginning of 2022, if we have a good vaccine, fingers crossed, we could be approaching herd immunity in some parts of the world. In some countries in East Asia, that could take much longer because very few people have been exposed to the COVID-19 infection in the first place. So you need to vaccinate a lot more people, and that process could take longer. But in some parts of, say, Italy or some parts of the US, where so many people have caught the infection by then, herd immunity with the vaccine could be achieved reasonably quickly.
I think globally people are very excited for the vaccine to really being rolled out, no? So you mentioned a while ago, I'm not sure if I have taken it correctly or not, but are you saying that once vaccines will be rolled out realistically, that the other public health precautions like, you know, masking up or distancing will still be in effect even if the vaccines are rolled out?
Yeah, I think so, because as I mentioned, it's very unlikely that we can vaccinate all the billions of people in the world at once. It's very likely that we're going to vaccinate at-risk groups like healthcare workers or the elderly to start off with, which means that there's going to be a huge proportion of the population that's still susceptible and unvaccinated. So some combination of the public health measures that we are currently facing is going to be ongoing, even if a vaccine should come out. I think there's very little doubt about that, especially in East Asia, because not enough people have been exposed so far. So a lot of people are still vulnerable and susceptible.
So we really have to put it in our routine to mask up when we're sick and distance ourselves, no, from one another for in the meantime. So there's a question from one of our participants here. So are there any promising interim medications while we wait for a vaccine?
Unfortunately, it's not looking good. We had a lot of hope for remdesivir from Gilead, and the WHO Solidarity trial has come out saying it doesn't really protect against mortality, that particular outcome. The medication that Donald Trump took, Regeneron, which is a monoclonal antibody cocktail, it looks very promising in very preliminary studies and in monkey experiments, but how it works in people, we are actually not certain of, and we're waiting for the phase 3 trial. I'm more optimistic of a vaccine rather than an antiviral, to be honest, because vaccines, we are good at making good vaccines, humanity in general, but we're not that great at making antivirals against respiratory viruses, to be honest. So it takes a long time to develop an effective antiviral. So I think the vaccine is what I'm more hopeful for compared to a drug. Yeah.
I think there's a follow-up here about the tos, tocilizumab. Are you familiar with the
Yeah,
medicine?
tocilizumab was a very
Are
exciting.
there any updates for that?
There was a very exciting approach in that theoretically they said, hey, COVID-19 causes a lot of inflammation. So if you reduce the inflammation, you can improve outcomes. As far as I'm aware, the company issued a press release saying that the phase 3 data was less optimal than they would have liked. So I don't think they've published the final data. But when the company says it doesn't work, I believe it really doesn't work. So because they have such interest in, developing and developing it. So I'm afraid tocilizumab, it might work in some selected settings, but it may not be the panacea that we were hoping for. Dexamethasone is one thing, the steroid, very simple kind of intervention for people who start requiring oxygen. There is very robust evidence that it works. Yeah. So I would say that the only effective antiviral we have to date is the steroids, good old steroids. Yeah.
Thank you for that.
Not exactly an antiviral.
So I think most of the medicines or most of the vaccines are now on phase 3, no? So a participant would like to ask if are there any like minimum requirements for the regulatory authority to grant authorization before anything else? There's the minimum requirement at least.
This depends on the quality of the regulatory agency. So we can see that some governments have chosen to speed up regulatory approval for vaccines even before you have any phase 3 data. And that is ridiculous because you simply don't know if it works. So why would you offer regulatory approval for that? But we've seen a lot of political pressure, unfortunately, on regulatory agencies to do this. But to answer your question in a more objective way, WHO is, I think the standard they have set is vaccine efficacy of at least 50%, which means that if you get the jab and there's 50% reduction in symptomatic infection, then you can say that you have an effective coronavirus vaccine. So that's the definition of efficacy set by WHO. So regulatory agencies would do well to examine the phase 3 data, look at the safety, obviously make sure there's no alarming safety signals, and also look at the vaccine efficacy. And if it meets the WHO requirement, then that would be a starting point for regulatory approval.
So I think the next question is again equally interesting. So they wish to know whether this is related to either reinfection or the capability of the SARS-CoV-2 to mutate. No. So the question is, since some people are reinfected or after recovering, the existence of strain mutations are speculated. How will this affect the generation or design of the vaccines? So will we need a multi-spectrum kind of vaccine for real protection?
It's possible. What we know about reinfection so far is that it definitely happens, but it's not super common. At least in the short term after getting infected with COVID-19, most people have short-term protection, at least as far as we know. Obviously, we can't see into the future. But some people have reinfections. And based on how many reports we are seeing, it's not something that happens particularly commonly. The reason they get reinfected is uncertain. One of the reasons is what you proposed, that is the strains are mutating in some way, so they're infected by another strain. But the other reason is perhaps because their immune system has some blind spots to the virus. It's not particularly well directed against COVID-19 for some reason, and these people then get reinfection. So really, it depends on the reason. Now, as far as the vaccines are concerned, it is possible that we may have to update the vaccines. So the second gen or third generation vaccines may need to have more than one strain. or you may need to change the strain that you're using in your vaccines. But coronaviruses in general do not mutate too much because their RNA polymerase is very clever. It actually is able to proofread as they go along the genome. So unlike influenza, where you have to keep reviewing every single year, I would expect the COVID vaccines to be a little bit more stable than that. So you would probably not have to keep changing too often. But time will tell. My expectation also is that in general, reinfections are going to be occurring more commonly in the long term after somebody catches COVID-19 the first time, but the reinfections may be less severe. So it's also possible that it's not something that is going to be particularly alarming. So I'm not too concerned about the reinfection issue. We're just looking for a vaccine to break the virus to most people for the first time. And then if and when they get reinfected, the hope is that the infection would still be mild. So that's kind of my speculation on that. So hopefully I'm optimistic as far as that is concerned.
So I'm trying to imagine like a flu vaccine that you have to take annually or something like that. That you need to.
Yeah, my expectation is that disease severity will go down. So it won't be cost effective to give it every year to all people, but we'll see.
Or just take the necessary precautions like masking up, right? All right. So I have the last question, but for the participants, both from Zoom and for Facebook Live, no, I think Dr. Sidar will stay naman for a while. He can type the answers to your questions in the chat box after this time. Okay. So for the last question, sir, what do you think is the importance for scientists like you who are directly involved in COVID-19 bench work, like for vaccine bench work, to be highly visible in social media or, news platforms such as webinars?
Highly important, I'd say. I think we're seeing a lot of misinformation, especially related to vaccines. And it's perhaps important to give people a more balanced perspective on what is going on. I would not say COVID-19 vaccines are definitely effective. They're definitely going to work and you must get it once they come out. I would also not say that they're ineffective or they're useless. These are very polarizing views. So I think scientists who are involved in COVID-19 vaccine development could give a more balanced perspective to introduce people to the complexities and nuances of vaccine research. And I think that voice can be best provided by people, vaccinologists working in the field. So it's a difficult subject. It's a difficult subject to communicate effectively. And the more, the better, simply. Yeah.
Yeah, that's right. So we really need to fight misinformation and translate scientific knowledge easily comprehensible for the public. So I'll be ending this open forum. Dr. Siddharth, thank you very much for answering our questions. I think there are some questions in the chat box, so I hope you could type in the answers later on. So I think at this moment, I'll be awarding you your certificate of appreciation. So let me just read the citation of your certificate. It's kind of small on my view. Okay, sorry. All right. So the certificate reads, the Philippine Network of Microbial Culture Collection Incorporated awards this certificate, in cooperation with Trinity University of Asia, College of Arts and Sciences, Art and Education, Department of Biology, presents the certificate of appreciation to our speaker, Dr. Siddharth Sidar, for I'm sorry, the font is really small on my screen, so I can't help. I can't read it. Let me. Can you make me small and the certificate big. Ayan. Okay, for his invaluable contribution as a speaker during the 20th PNMCC Annual Symposium Webinar Series, the 3Cs, Culture Collection and COVID-19, given on this 17th day of October. This is signed by our President, Diane Dizon, and of course the Dean of the College of Arts, Sciences and Education of Trinity University of Asia, Professor Howell Hall. Thank you, and again, congratulations, Dr. Siddharth Siddhar.
Thank you so much for inviting me. Thank you. It's been a pleasure.
Thank you very much. Okay, so I think we are now moving on to our next speaker. Our next speaker will also be talking about vaccines for COVID-19, but now there is a local touch for it. So let me introduce to you our speaker, Dr. Nina G. Gloriani. She is a retired full professor of medical microbiology and immunology. Currently, she is a clinical microbiology consultant at the St. Luke's Medical Center, the chair of the DOST Expert Vaccine Panel on COVID-19 vaccine trials, and a member of the WHO Scientific Steering Committee for COVID-19 Solidarity Vaccines Trial. She is an MD with a PhD trained at the UP College of Medicine, and her PhD at the University of Melbourne, Australia, taking in microbial immunology and immunochemistry. From 2007 to 2013, she served as Dean of the College of Public Health, UP Manila, and concurrently, the Director of CIMEO TropMed Regional Center for Public Health, Hospital Administration, Environmental and Occupational Health. For several years, she also served as the director of the Institute of Biotechnology and Molecular Biology of the National Institutes of Health, and also chair of the Department of Medical Microbiology of the College of Public Health. For these positions, she established the Microbial Immunology Research Group, which addressed sero-epidemiologic and molecular studies on a number of infectious disease agents, including hepatitis A, B, C, and E, HIV, herpes, and Hantan virus. And from 2010 to 2015, a very significant contribution. This is the 5-year JICA DOST-funded program for the prevention and control of leptospirosis in the Philippines. Their team have developed the Leptovax vaccine against 4 leptospirus serovars that are prevalent in the Philippines. So her main research interests are vaccine development and clinical trials, made her very fit for this talk this morning. And also development of diagnostic kits and studies on host immune responses against bacterial and viral infection. She is a highly prolific researcher and academician, and a very supportive and loving mentor to whom I get my love for virology and immunology from. Please, everyone, help me welcome Dr. Nina G. Gloriani for her talk entitled Vaccine Platforms for COVID-19, Which Ones to Choose and Why. Dr. Gloriani.
Yes, good morning, everyone, and thank you very much, Dr. Marilyn. She was my student, but now she's a better professor than myself. She has gone so many beyond my expectations. So thank you, and to Dr. Professor Dayan, thank you for inviting me here also. And I'd like to take this opportunity to say hello to the president of Trinity University Asia, my longtime friend, colleague in immunology, Dr. Wilfred Chu. I'm happy to see him, although virtually. So I'll share my slides now. Okay. Thank you very, very much, Dr. Siddharth, for the very comprehensive discussion on the vaccines and COVID-19. You have paved the way for my slides, my talk this morning. So my title is Vaccine Platforms for COVID-19, which ones to choose and why. There may be some overlap, but for the most part, I will be presenting the Philippine Solidarity Trial under the WHO supervision. Okay, this is my outline. Well, first, I saw Dr. Sidhar say about conflicts of interest. I declare no conflict of interest. I am now retired and I am not with any private company. So, the outline for my talk, WHO COVID-19 vaccines landscape. This is the as of October 15. COVID-19 vaccines platforms, the older technologies versus novel vaccines. So I may dwell into some detail, but I'll try not to be as very technical. I was requested to put in a little about reference cells and virus strains, which I'll do. I think I have about 3 slides there. And we'll give you an idea of what's going on about clinical trials for COVID vaccines here in the Philippines. So the global perspective and the Philippine perspective, and then some take-home messages. Okay, I think you have seen this, except that my date is October 15. So this is from the New York Times. I don't know why New York Times and WHO have different data, but on this slide, the left side panel here, note here that we have around 42 COVID-19 vaccines under phase 1, 2, or 3 trials. And note that most of them are protein subunit, but we do have inactivated and non-replicating viral vectors, which Dr. Sidhar Hart already said are the primary ones that are now in the advanced stages of development. So I think he went through all this just to make this point that there is a very long process before we can get the vaccine to the public. Okay, so these are the vaccine platforms. Just to maybe reiterate for some of you, we have this whole virus here that may be made into a vaccine by inactivation or attenuation, or just work on these very important domains, antigenic domains, specifically the spike proteins of the SARS-CoV-2, and make them into vaccines. So I just placed here 10 candidates which are in phase 3 trials as per the WHO landscape. And actually, we have been looking at this at the vaccine expert panel level of the DOST. So we have some information on their technical dossier. So for the RNA vaccines, the leading candidates are from Moderna and Pfizer, Biotech, and which is also collaborating with Fosun. So these are from USA. There is this vaccine from Novavax, this DNA vaccine also from USA. And the vectored vaccines are from 4 companies: AstraZeneca, Oxford, Gamaleya. I think you hear a lot about Sputnik V. So we have had a chance to also evaluate that vaccine. Then Janssen is also applying here in the Philippines, but CanSino is not. Then for the inactivated vaccines, we have Sinovac, which is applying for a clinical trial here in the Philippines. But Wuhan, the Sinopharma has 2 strains, Wuhan and Beijing inactivated vaccines. But they, I think, will just be involved in distribution, but not clinical trials. Okay, so this is what I was saying. No, there's a lot of detail here, but just to maybe differentiate the 4 major ones, actually 4, no? Kasi ito yung whole virus, then you have the nucleic acid, which could be DNA or RNA, the protein-based, which could also be used synthetic peptides, but for the most part, these will be recombinant protein-based vaccines and the viral vector-based vaccines. So how it works, you use the whole virus after it has been inactivated or killed with heat or chemicals. Here, you destroy the RNA or DNA, so it cannot replicate anymore. That's why you kill it. And what do you see in terms of immune responses? Having killed it, there is usually robust immunity against most parts of the virus, but these antibodies may also wane over time, and for inactivated vaccines, boosters may be needed. Usually, they would elicit antibody-mediated immunity, but not cell-mediated immune responses. So again, we have two which are under phase 3, Sinovac and Sinopharma, two from Sinopharma. So for the live attenuated vaccine, we do not have any information on this, at least in the Philippines, although you can probably see them online, some of their technical data online. This is a weakened version of the actual virus and will not cause disease, but can still replicate and stimulate an immune response. Because it is live, there is longer antigenic stimulation and it may produce long-lasting cell-mediated and antibody-mediated immune responses. So you have both CMI and humoral immune responses. So there is a Codagenics Indian company that takes care of this, that develops it. Then for the nucleic acid based, we have actually more of the RNA vaccines than DNA, but it says here it uses DNA and RNA to teach the immune system to target key viral proteins. So these sequences would encode instructions for making the spike proteins, which, as has been presented earlier, are the ones that we need for neutralizing antibodies. For the DNA vaccine, these are usually bacterial-derived DNA plasmids modified to include a gene that codes for a viral protein such as the spike. Now, for the immune responses, these are, of course, against the specific viral antigens. In this case, they encode for the spike, especially when delivered with lipid nanoparticles. So here we have RNA vaccine from Moderna and Pfizer, and Arcturus is collaborating with National University of Singapore. And then we have for DNA Inovio. So we have also a chance to actually evaluate this. Then for the protein-based vaccine, this is a piece of a virus surface. So they are actually working on the protein itself to focus the immune system on a single target. And instead of the nucleic acid, you directly deliver the specific spike antigen into the host. So subunit vaccines generally only induce antibody-mediated immunity, not cell-mediated immunity. So there, you will also need boosters a lot. And for this, we have Novavax and Medigen. I think we have some data on both of these vaccine candidates. And we have here the viral vector-based vaccines. They use weakened live versions of safer viruses like adenoviruses as carriers of genes encoding for COVID-19 spike proteins. So the host cells will make copies of the spike proteins, and the adenovirus activates the immune system. And in so doing, will also activate the immune response against the adenovirus and the spike proteins. So these viral vector-based vaccines are good at stimulating immune responses, but still largely experimental. So we have two candidates here that we have had the opportunity to evaluate a little AstraZeneca and more about Janssen. Now for the pros and cons. Now these two having using the whole virus are actually what we call older, trusted, maybe older tested technologies. We have a lot of information on their safety profile, efficacy, maybe immunogenicity. They are, at least for the inactivated form, it is harmless, but still recognized by the immune system. And we have many facilities across the globe have existing manufacturing plants or facilities that are capable of producing these inactivated vaccines. The final product, generally more temperature stable and storage conditions are available, for instance, here because they would require only refrigerator conditions. But cons, hard to make. You have to grow the virus in massive amounts. Therefore, you have safety issues when you are dealing with the live virus before you can attenuate them. And there is also a slow process and not easy to scale up. Not that easy, although a lot of the bigger vaccine companies all over the world have establish bigger facilities in order to scale up, especially in China. The live attenuated vaccine, as already said, is able to replicate and provide long antigenic stimulation. There is also existing manufacturing capacity available and relatively cheap to produce. But as I said, you have to grow the virus in large scale. It takes a long time to weaken the virus to a level that will make it safe enough and yet still stimulate the immune response. The possibility of reversion to wild-type virulent virus mutation is there, and most of them would require minus 20 degrees storage. And of course, we know being live, they will not be suitable for the immunocompromised. Now, for the nucleic acid-based vaccines, now these two, nucleic acid and viral vector, the what we call under the next generation or newer novel technologies for vaccine development. So relatively, it's fast and easy to make these RNA or DNA vaccines once the genetic sequence is known. That is how Moderna was able to produce its messenger RNA vaccine in record 2.5 months, I think, from the time that the Wuhan strain was sequence was made available to the public. So it is relatively easy to manufacture millions of doses quickly. And right now, a few proprietary platforms have shown it to have higher stability for the nucleic acid-based vaccines. However, there is no nucleic acid-based vaccine available to date commercially. It, so it remains largely unproven, still largely experimental for people, but among in animal tests, they have performed well. And we all know that RNA is less stable than DNA, and more that temperature instability would require minus 80 degrees centigrade storage conditions. Although there are some platforms that we read that have improved, stability conditions, storage conditions. Now, for the protein-based vaccine, many scientists are familiar with this. That is why it is the most um, popular one that is being developed now. And it works well. We know the human papilloma virus vaccine and the hepatitis B vaccine are based on the recombinant DNA technology, considered very safe and less likely to trigger side effects. So, but cons, the small piece of virus may not be enough to elicit a strong enough immune response, even with adjuvants, and usually would need multiple doses to build up enough immunity. And for the viral vector vaccines, living but weakened virus, like the adenovirus, would stimulate a robust, durable, and long-lasting immunity. Actually, the SARS-CoV-2 vaccines that are patterned from the use of adeno, took off from the experience in the Ebola vaccine development, which is currently available under emergency use authority. Although, of course, there is no Ebola outbreak right now, but that is available under EUA. So it's still an experimental platform. Concern, this was already discussed earlier, that some people may be immune to adenoviruses and render the vaccine ineffective in those individuals, so will not work for them. So in anticipation that some of these viral vector adenovirus-based vaccines will come in for COVID here in the Philippines, DOST will be um, doing a study on the seroprevalence of antibodies against adenoviruses among our population. Okay, so a little on the cell cultures and strains. So before you can actually make any vaccine, of course, at least for the inactivated and the live attenuated, you need cell lines. So I'll just mention a few of these. So we have vero cell lines in vaccine production. The vero cell line is derived from the African green monkey kidney, has been widely used for viral vaccine production. And because for many years, these cells have been known to be sensitive to infection with so many viruses like measles. So these viruses, arboviruses, polioviruses, especially the poliovirus, measles, they are grown in vero cell lines, influenza virus. And now we're looking at coronaviruses as well. And the virus cell line is also used for the amplification of several SARS-CoV variants. Now, I'd like to refer you to the WHO Viro Reference Cell Bank, RCB 1087, which provides a unique resource for vaccine development. You know, you need standard, stable cells to grow your virus so that you will have the vaccine you produce will be also standard. Cell substrate with a safe and reliable history of use. I think this came about because of certain issues with the cell banks before. So they have to standardize this and under the auspices of the WHO. So this cell bank, reference cell bank, provides well-characterized cell seed material for generating a master cell bank that vaccine developers could use. It is available free of charge on application to WHO and currently stored in the American Type Culture Collection and the European Collection of Animal Cell Cultures. Now, something that came up also recently is that of the virus STAT1 knockout cell lines. ATCC used gene editing, so CRISPR-Cas9 gene editing technology to develop these knockout cell lines that can produce high titer viral stocks for vaccine development. So you want cells that will produce high titers of viruses. So these cell lines are also able to package lentiviral, adenoviral, and retroviral vectors. So you also need that. We have heard about the vector-based vaccines for delivery of DNA sequences into target cells. So so you see here, this is the parental, this is the knockout one. So it has more of the viruses lighting up there. This cell line has a homozygous 199 deletion spanning the third and fourth exons of STAT1 gene. And this cell line, as I said, exhibits significant increase in viral titer, as shown here, versus the wild type, and enhanced virus production capability compared to its parental line. So I think that is available from ATCC. Now that is for the cells that will allow growth of these viruses, though you also need reference strains for the viruses, and ATCC has authenticated and fully characterized reference strains that could be used for many purposes. So you know that you have to select virus strains for vaccine development that must show consistent immunogenicity, induction of resistance to reinfection, and low rate side effects. So they have to be consistent. They have to be well characterized. And these are just some of these reference materials available in the ATCC. At least two are fully sequenced reference materials from the US. These are from the US. Hit inactivated, and they also have genomic RNA preparations available from the ATCC genome portal. So either from Germany, they have many different isolates that could be accessed. Now, what are the uses of these strains aside from vaccine development, of course? So having reference strains to test antibodies, for instance, will be very crucial in coming up with very accurate results. This test serum, for instance, collected from people who have recovered from COVID-19 to detect antibody that may block viral infection will be tested against reference strains, or at least the tests that become available will have to use reference strains that are fully characterized. So for other research uses, of course, for antiviral research, you have to use standard well-characterized strains test ability of existing or experimental antiviral medications to treat SARS-CoV-2 infection. We know that you need very well-characterized strains for developing inactivated or live attenuated vaccines. You need these strains to do pathogenesis research. For instance, determine ways by which virus can be transmitted to a host, the severity of illness, viral load, or the organs targeted by the virus, and even for virus stability research, which may indicate how long the virus can survive and stay infectious under different conditions, temperatures, where they can survive. So all of these are applications. Okay, so now we move on to the vaccine candidates and the approval process, at least here in the Philippines. Whatever the platform technology used to generate the COVID-19 vaccines, whether old or new generation technologies were used in vaccine production, the investigational product has to satisfy standard criteria and pass stringent tests. So from vaccine discovery to preclinical tests in vitro and in animals and finally in humans. So actually there are other bodies that will need to approve the vaccine either for clinical trial, but for the registration itself, the FDA Philippines will have to approve it. Okay, so this was run through somehow earlier by Dr. Sidharth. So just take note, we need data from preclinical vaccine development and up to phase 1 and phase 2 data before anybody can conduct phase 3 trials in the Philippines. So the vaccine expert panel evaluates all of this preclinical data, tests for safety, efficacy, proof of concept validated, potency assays, everything. And moving into phase 1, they should have safety data, local systemic reactions, some immunogenicity. Usually there is a combined phase 1, phase 2 trial under the outbreak paradigm of vaccines in COVID-19. So immune response for phase 2, safety also, optimal dose and the schedule of giving the vaccine. Of course, always safety assessment is there. So I want to reiterate that only vaccines that have completed the preclinical and clinical phases 1 and 2 trials will be evaluated or are currently being evaluated and approved for the conduct of phase 3 trials in the Philippines. So the vaccines are usually tested for three things: safety, immune responses generated, and efficacy. So it depends on the phase you are in. You're looking at local and systemic side effects, normally mild, but there could be adverse events and serious adverse events in some rare instances, and they all have to be monitored. Then immune responses generated, we're looking at neutralizing antibodies and also correlates of immune protection in cell-mediated or T-cell responses. And lastly, the efficacy or protection against COVID-19 disease. And Dr. Sidharth already referred to different, indicators of efficacy. I will explain for the Solidarity vaccine trial what we're looking at for efficacy. Okay, so phase 3 only. We do not do these other phases here. Whoever wants to do clinical trials in the Philippines will have to complete all of this up to phase 2, results of their phase 2 trials. So phase 1 is only a few people tested, phase 2, hundreds to maybe 1,000 volunteers, and phase 3, thousands and thousands of volunteers. And then after everything is deemed okay, all the data show that it is safe, immunogenic, and immunogenic. At least we are not looking at efficacy yet here. That we can proceed with the phase 3, and if the phase 3 trial shows efficacy, safety in target populations, then the vaccine could be registered by the FDA. But after registration and administration to the population, there should be post phase 4, which is post-marketing surveillance for rare adverse events as vaccines are rolled out in the general population and in small groups. So we have the two types of vaccine trials in the Philippines. The first one is that of the WHO Solidarity Vaccine Trial, supported of course by WHO and then by the Department of Science and Technology and the Department of Health. The Philippines has officially submitted its intent to participate in the Solidarity Trial and has nominated the principal investigator to oversee the Solidarity Trial. This is Dr. Jodor Lim from the Philippine General Hospital. But we are also looking at independent clinical trials to be conducted by bilateral partners, Sputnik V from Russia, China, Sinovac, and then the rest are some how are in early phase trials, but will also but are also planning to conduct clinical phase 3 trials here in the Philippines. And there are other private vaccine companies. These bilateral partners, we have CDA agreements with them. Okay, so what is the Solidarity Vaccine Trial? I just want to highlight is phase 3, okay? So it is an international multi-center, meaning so many sites around the world, double blind, meaning the vaccine participants do not know what they are receiving, whether they are receiving vaccine 1, vaccine 2, vaccine 3, or placebo. And the one administering the vaccine also do not know what they are giving, what he is giving to the recipient. So this is a placebo-controlled randomized trial of candidate vaccines against COVID-19 because it is multi-center. There are many sites, more than 200. I think they're looking at more than 250 trial sites around the world have expressed interest in participating, and that includes the Philippines. So there are 6 regions under the World Health Organization: Africa, America, Eastern Mediterranean, Europe, Southeast Asia. We are under the Western Pacific region here. So I'm putting the map of the Philippines because we are proud to be participating in this trial. The government is setting up a lot of money for this trial. Okay, just a little explanation on the whys and the wherefores, the whats of the about the Solidarity Vaccines trial. So as I have said, this is an international randomized trial of several candidate vaccines. So 4 things here. The Solidarity Vaccine Trial will evaluate several different candidate vaccines quickly, and that will increase doing that will increase the likelihood of finding several effective vaccines. However, the vaccine candidates to be tested are not yet identified as of today. So we are hoping by within the next few days we will know which vaccines will be under the Solidarity trial. Then, as well as evaluating several different candidate vaccines quickly, we are expeditiously enrolling participants at sites with high rates of COVID-19. And thus, we can have rapid accumulation of data to support rigorous evaluation of the candidate vaccines. This will also eliminate the inefficiency of designing and conducting separate trials. If you conduct separate trials, you will require thousands, maybe 30,000, 40, 50, 60,000 subjects or participants. Now, so all of this would have to get results within 3 to 6 months after each vaccine is ready for inclusion. And the last advantage to this is that the international collaboration and countries' commitment will foster international deployment, hopefully with equity of access to the vaccines, because availability of the vaccine does not mean it will be accessible. So we hope that by participating, we will have access to the vaccine. So just a little on the global framework, the trial governance framework. This is a supervised by a global international team. This is the Scientific Steering Committee, which deals with the WHO leadership, and I am a member of the Scientific Steering Committee. There is a global data safety monitoring committee that will report to the steering committee the progress of the vaccine during implementation and whether there are adverse effects and other critical data that need to be taken up. Then there are, of course, co-sponsors. The member states become co-sponsors, plus the funders of the WHO. Then the global trial team. There is a global trial team to whom the local teams report to. So at least once a week, there is a meeting of the different local teams with the global trial team. Just for you to understand how much documentation we require. So for any of these clinical trials to proceed, The WHO Solidarity Vaccine Trial documents and SOPs are actually listed here. There may be some others that I may have missed, but we require a very uh, comprehensive protocol. We right now have an expanded protocol from the Solidarity Trial, but we are still waiting for the final one. The very detailed statistical analysis plan, the SOPs, which you see here, include all of these. So all of these will have to be submitted, like what endpoints. The primary endpoint for the Solidarity vaccine trial is COVID-19 virologically confirmed case, because there will be secondary endpoints that could also be ascertained, how we enroll the participants. Informed consent form is very important, and which the ethics boards will require. And we have information, randomization, and blinding, and so on and so forth, including laboratory procedures, blood samples processing, blood sample collection, of course, very important, the adverse effects management and reporting, or severe adverse events management and reporting. So all of this will have to be submitted. Now, there are guidelines for for preparing under the global participatory practices. And just to show here, before the trial starts, which we hope will happen sometime soon in November, we should have, as a participant, we should have developed a succinct and evolving stakeholder engagement plan, operational communication and issues management plan, and stakeholder engagement plan in all aspects of trial preparation. And for this, we have the Philippine Department of Health, through the Health Promotions Bureau Communications team, planning all of this. And I think they have available in various social media platforms already, information on the solidarity, and mostly on the solidarity vaccine trial here in the Philippines. They will have a portal with the Philippine Information Agency that you can access, anybody can access anytime. There are also platforms where you have question and answer, and these are actually updated weekly. Then, for the setup of the optimal trial conduct, study research team, coordination and communication structures, administrative and logistic systems, the Philippine Solidarity Trial vaccine teams, together with the local government units, are the ones preparing for this. So we are at that phase before the trial relaunch. Now, for a brief overview of what we actually will be doing, so the WHO Solidarity Vaccine Trial is an adaptive trial. High transmission areas are prioritized for vaccination. We call them trial zones. Now, why high transmission areas? Of course, this is an efficacy assessment. So we want to know if those who are vaccinated will be protected from the COVID-19. And you cannot do that if there are no cases where they are exposed to. So under this, we have the trial zones. We have fixed trial sites in hospitals, or we can also identify some community health stations provided they meet certain criteria. But an additional special feature of the Solidarity Trial is that we can have or set up mobile trial sites or pop-up sites. So this is based on the concept of ring vaccination as first expounded with the Ebola vaccine. So what is ring vaccination? So this is your ring. Ring vaccination is an infection control measure that involves vaccinating a cluster of individuals at high risk for infection based on their social or geographic connection to a known case. So if you have the case here, those around the case will have to be vaccinated. The ones closer, and maybe a third layer, but maybe up to second layer only. So this should create a protective ring or cluster of immune individuals around newly diagnosed cases, preventing further spread of infection. So that is what we want to happen. So we are preparing very hard, because it will not be easy to set up these pop-up sites. The fixed sites, it's okay. The community health stations maybe will have more challenges, but the greatest challenge will be with the pop-up sites. Now, for the whole Philippines, we are probably going to contribute 1,500 to 2,000 participants for the country alone. Now, for the entire world, 20,000 participants per vaccine, and then for 20,000 for a comparator placebo. So we may increase this number of participants to 4,000 depending on the supplemental budget that we will get. We are still requesting for the supplemental budget. So the priority group to recruit is 18 to 59 years old, high risk for COVID-19 infection, such as the healthcare workers, frontline workers, and as I have said, the contacts of cases. But special groups may be included later, such as the elderly, 60 years and above, children, immunocompromised. As we all know, of course, inclusion and exclusion criteria will be defined. So when we go to the communities, they have to understand that only people who satisfy the inclusion criteria will be able to participate. So inclusion criteria means they should be healthy, they should have no COVID, they should be able to adhere to the monitoring and the follow-up procedures. And exclusion criteria, generally, if you have had COVID, you are excluded. If you have unstable, if you have other diseases and you are unstable. But if you, in some areas, for instance, among the elderly, if they are stable under certain medications with some protocols, they may be allowed to be included. And randomization of vaccines, we do not know yet how many vaccines under the Solidarity trial, but randomization will be made by WHO. The immunization schedule could be 1 to 2 doses. Currently, only a few vaccines will use 1 dose. Most of them will use 2 doses. So either with a 14 to 28-day interval, depending on the vaccine type. So that is why we're closely monitoring these vaccines that could enter the Solidarity trial. And we know that vaccinated individuals will be monitored regularly according to the SOPs that I mentioned earlier. So for the Solidarity trial, the primary outcomes should be available within 3 to 6 months after immunization. And I already said that the primary outcome here is virologically confirmed COVID case in the vaccinees or those who receive placebo. But secondary outcomes like immunogenicity, safety, and yung other efficacy measures like from severity, COVID, even death, as participants will be followed up for 12 to 18 months. The support that the DOST is giving to the Solidarity trial in the Philippines will last up to 18 months. So which trial sites were chosen for the Philippines? So remember I said we need high transmission areas. So NCR is the highest among these regions here. So also we are in NCR, so it is easier to monitor, to coordinate immunization implementation here. So we have clustered the different tertiary hospitals shown here in different colors. Cluster A, Manila area, that's large Manila area. PGH and Manila Doctors Hospital will lead the vaccine implementation here. Cluster B is the blue one for San Lazaro Hospital. Cluster C is for Lung Center of the Philippines and St. Luke's Medical Center QC. Cluster D is the violet one here, RITM and Makati Med. And Cluster E, the Medical City with St. Luke's BGC. Okay. There are other potential sites in Cebu. At the time that we were preparing this, Cebu had very high transmission rates, but now that has quieted down. But they may still have some cases that they can include. Cavite, we're still waiting for some documentation from them that we require. And Davao has already said yes to actually participating. So we're looking at that. Plus, we will also include regions 6 and 8. This is Iloilo and Bacolod because they are saying that they have high local transmission of COVID in those areas. So it will be a little more difficult because they were not initially included, but we will try our best to include them. So vaccine teams will have to work very closely with communities at the barangay level. So what do I mean by barangay level? because we will be working with the highest, the areas, the top 5 to 10 barangays with high COVID-19 case rates. So this was generated back in September 1, but it has been updated already, but I have not seen the actual map. So I just showed the September 1 data. So DOH Epidemiology Bureau provides us the data for the epidemic of the COVID-19. So this is your cumulative cases here. The redder, the more cases you have. So these are the active cases and these are the attack rates. So generally, we will be looking at attack rates, but we're also looking at consistency from maybe 3 to 4 weeks, consistently high transmission rates in these barangays. So right now, the hospital sites have submitted their top 5 to 10 barangays, and we are working on them for the inclusion as trial sites. Now, to give an overview of the trial process, this has already been discussed in vaccine teams by the global trial team. This is very much technologically assisted or supported. Now, under COVID-19 conditions, under quarantine conditions, group information or getting the potential volunteers or participants to come to an area will be very challenging. So we have to work hard on, social distancing and making sure that we follow strictly the health precautions. So there is this group information on the informed consent form, maybe 10, 10 people per in the morning and 10 in the afternoon. So not too many at any time. That is how much the vaccine team can handle. There will be trial ID and pre-screening, informed consent, signing, inclusion. Well, after that, I think it should be inclusion, exclusion criteria. Then biometrics, they will use the iris technology. So we know that we are dealing with the same person when they come back, when that person comes back for the second dose or for follow-up. So all of these will be barcoded, use of digital iris technologies, electronic CRFs, electronic data capture, barcoding, vaccine placebo randomization, and monitoring. So a lot of this will be provided by the WHO, and everyone in the team will be properly oriented in doing this. Now, are we ready? So this is one of the vaccine expert panel mandates. So we have to make sure that the Philippine team is ready. So we have these sites readiness milestones that we need to follow. So local sites PIs designated. Actually, we have done this even before August. So we have designated, as I have said, Dr. Jodor Lim of the Philippine General Hospital. So we're looking at sites context and experience, looking at, what they are capable of, their accreditation, and so on. Then we have to submit the epidemiological information, which we have been doing for the last 4 or 5 weeks. So that should have started in week 1 of September. So local disease incidents, including deaths. We have very minimal zero prevalence studies, but we hope we can get more from the private companies, private institutions who perform these zero prevalence studies as we go along. We provide that. We're putting in place the SOPs. We do have, even without the informed consent from the global, we're preparing our own that are site specific. So we have all of this being ready for the trial and even the laboratory requirements. So lab capacities, SOPs and tests who are in charge for the RT-PCR, this is an efficacy endpoint. So we want to make sure that all of these hospitals that we actually identified will be able to perform the RT-PCR test immediately upon receipt of the specimens from the vaccinees who will develop signs and symptoms. Initially, it was thought that the pilot run of the Solidarity trial with 1 up to 20 countries could start by the 3rd week of October. That there has been some delay because of the identification of the vaccines. They have not identified the vaccines. I do not know what is delaying that, but we are looking at a delay of about 3 to 4 weeks. I hope not longer than that. So maybe we are hoping we could start the solidarity trial. If not the pilot, we can join the other selected sites a week after. So maybe November, end of November. Hopefully not during the Christmas season, that December. And because of that, the interim results we expect maybe by February. So I think somebody asked about approvals earlier. Just to make it clear that there are many levels of approvals for these vaccines to reach the market. So for the start, even for the clinical phase 3 trial, the Philippine-based clinical sites, clinics, hospitals, pop-up sites in communities, they have been identified and they have to submit the requirements like the accreditation for COVID-19 testing facility, FREB Level 3, Research Ethics Board's accreditation, the CVs of the principal investigators, their current PRC licenses, so they have to submit all of this. So we have here all of this, and as I said, we can include other hospitals as needed. Then the ethics review boards also have their own requirements. The informed consent form is very important. There are site-specific requirements also that the vaccine trialists have to consider submitting. Now, to all this, WHO has target product profiles and all SOPs that need to be followed. The target product profiles could be accessed from their site, and actually one of them was mentioned by Dr. Sidharth that at least a 50% efficacy is required of these vaccines to actually be included or considered for approval. And lastly, but most important, not the least, but the most important is the Philippine FDA approval for the clinical trial. And later, when the clinical trial has been finished, all the data should be coming in again. Then it will be registered and licensed as vaccines for rollout into the general population. So again, I say we're talking about a timeline of between November to April 20 for the primary endpoints, and for the secondary endpoints, maybe up to early 2022. And that we have your solidarity trial, but the other vaccine developers will go through the same process. So here, this is where the vaccine expert panel works before these other things here. So my last slide here, take-home messages. You have heard about multiple approaches towards the same goal of producing a safe, effective vaccine against COVID-19. But it is very likely that different vaccines, not just one type, will be needed for the pandemic response. You have seen their advantages and disadvantages in terms of immune generation, capacity for scale up, and so on, safety features. And multiple companies with different vaccine candidates must work together to meet the global demand. Is it older technologies versus next-gen platforms? Most likely it will be a combination of both. And while fast-tracking vaccine development efforts, we need scientific rigor and integrity in approving vaccines for licensing and commercialization. And not to forget that we have to monitor what goes on after we roll out the vaccine. So post-marketing surveillance for safety and impact to public health. So I come from public health, and there is no intervention that we just leave as an intervention without looking at its impact to the general population, especially to public health. So that's it. Thank you very much for your attention.
Thank you very much, Dr. Gloriani. Can my video be on? Anyway, Dr. Gloriani, again, thank you for that very interesting talk about the local flavor, no, of the vaccine trials. I'm sure many of the participants right now are also excited, no, to know, no, the results of this particular trial. So I have here collected some questions from both our Facebook Live, from our Zoom, no. So my first question, ma'am, is that you mentioned about this trial or phase 3 trial being ethically approved, etc., and also, and it would involve thousands of participants. So if they will be recruited, no, or invited to participate in this trial, and if there are anxieties, for instance, can they say no? Do they have the liberty to say no, ma'am?
Oh, of course, under the guidelines yung sa ethics review, if they already said yes, they can withdraw anytime after that. That is included in the informed consent. So they will come during the general orientation. If they are not convinced that they have to participate, of course they can say no. Nobody, this is volunteer. No, nobody will be coerced.
Okay, that's right. Okay, thank you, ma'am. I think we have here two very interesting questions from Dr. Wilfred Chu. So the first question is that, are laboratory human immunogenetic data being collected for future analysis of vaccination results? That's the
Yes.
first question lang naman.
First muna, yes, baka malimutan ko pag dalawa. Yes, Dr.
Yes,
Chu.
yes, ma'am.
Yes, actually, part of the informed consent will include consent to the use of their blood samples and the nasopharyngeal subsamples for future researches, because we're already planning for the secondary endpoints. So like the duration of immunity is not part of the endpoint for the solidarity trial, at least the first 3 to 6 months, but it is of course part of the secondary endpoint. So we want to also sequence the isolates that we get, especially from both from, if we have isolates from vaccinees who received the vaccine and vaccinees who received the placebo. Later that will be unblinded. So we want to compare the sequence of their isolates, SARS-CoV-2 isolates in the future. So all of these participants will have baseline samples taken and stored. And along the way, if we have different timelines or milestones for collecting blood or even nasopharyngeal and oropharyngeal samples, they will be stored. So biobanking is a great part of it.
So that would be interesting that we'll have those samples biobank for future, no, considerations, especially for research, no, ma'am. And the second question of Dr. Tu is that, what studies will be done on those who suffered from mild symptoms and those who have recovered, in relation perhaps to vaccine development or other purposes, ma'am?
Well, we're all looking at the antibody levels, T-cell responses, and even transmission patterns. So that part on transmission patterns is a little more challenging, because the logistics involved in following up vaccinees, and then how he may be able to transmit COVID-19 to his contacts in the household or around, will be very difficult. But that is part of the list of topics we already planned for the future researchers. If you have more, you can actually let us know, because you may actually be involved. So we're just making this available to everyone, but we need very good proposals for the DOST-PCHRD to fund it. It will be a separate funding from the Solidarity Trial.
Oh, okay. So thank you, ma'am. So the next question will be coming from Eldrin, no? Mr. Eldrin Arguelles of Biotech UPLB. So he said that many individuals are hesitant about receiving COVID-19 vaccines. So reasons include the novelty and rapid development of the vaccines, as well as the politicization of the pandemic and inconsistent messages from either scientists or government leaders. So his question is that how do the Philippine government, together with the WHO Solidarity Vaccine Trial Team, no, address this issue, ma'am?
Okay, I think I may have mentioned yung sa isang slide, DOH Health Promotion Bureau. So very early on, I think as early as June, we were already in touch with them. So everybody, may core communications team. Sorry, hindi ko siya I was not able to present that slide
Yes.
on what they have prepared so far. But there is a parang plan, hindi plan, ano bang tawag doon? The main theme there is one voice, meaning one voice from the highest up to the down, from the president, office of the president,
Yes.
to the regional offices, to the mayors, the barangays, the scientists, our international partners. So they are into that. So they also have pre-launch, launch, and post-launch activities. I don't know how much of what they have prepared is already online, but I know that there are at least 4 communications materials that have been approved already. These are the frequently asked questions and answers, the primer on the vaccines, primer on the vaccine trials, and I forgot the other one. But time and again, I get consulted on to finalize we are consulted to finalize the messages in such communications materials. So they will be available, and I referred to a portal. I'm not sure whether I have not actually checked it. That will be uploaded by DOH HPB together with the PIA, the Philippine Information Agency, and that is where all of this will be also placed. So that will be accessible to all. So we also do not want media to be asking us every time. So what is there is, materials that have already passed the experts, the different groups. So we hope we can have that one voice as DOH envisions. So we deal with that way.
Uh-uh.
Para hindi, so
So ma'am,
I know politicization. We've seen a lot of it. We avoid that.
yeah, yeah. I think, ma'am, this is also related with the dengue vaccine scare of the people. No? So, for instance, there will be a chosen vaccine already that passed the phase 3 and then the regulation. Do you think there will also be an equal or parallel rolling out of, say, promotion and education campaigns about how this vaccine is really safe, is really something like that?
Yes.
Is it included in the
Oh,
One
yes.
Voice?
Yes. So I'm really sorry I did not include that one slide. Actually, it's a very comprehensive plan, and it has been rolled out already. So the DOHHPB communications team already met with the higher ups, the leaders, the heads, and then when was that? Just 2 or 3 days ago, we presented to the NCR mayors, they being the first, participants in this trial. So they are more or less now aware of what will happen, but the materials will have to be trickled down to them, and they will have to reproduce these materials because the DOH cannot produce it for everybody, but they will be available in social media, so anybody can actually access this. So
So that would be good, ma'am, kasi of course, the information should really go down to the barangay level, no, to the household level for that purpose.
yes, and may I add, when the trial starts to be implemented, there will be, even before that, we're planning on a dry run for the vaccine recruitment in a town hall like scenario, maybe in one of the barangays here in Metro Manila, in NCR, yes, to precisely to see how that will go, what sort of questions will be asked, and how many people will be coming in,
Yes.
how many people will say yes, I want to participate, because there is that hesitancy that we are also preparing for.
Uh, mm, that's right. Thank you, ma'am. I think I have the last question for you this morning. How is transparency being practiced here in the country for the phase 3 trial? Will they be open also, just like what happened to Johnson and Johnson when they had an adverse event? How do you investigate adverse event in this kind of trials?
Okay. So in addition to the Global Data Safety Monitoring Committee, we have the local boards to take care of monitoring the actual implementation. Okay. The VEP is no longer involved in the monitoring. We do the evaluation, which vaccines will be good for clinical trials. But upon implementation, three bodies will be there. The main monitoring board is the single joint reb and the site-specific research ethics committees of hospitals. The second monitoring board is that of the FDA. During implementation, FDA has a committee that also monitors what's going on in these different clinical trial sites. And there are endpoints that have to be reported. Of course, the safety, that is all spelled out. And lastly, the Department of Science and Technology appointed a local data safety monitoring committee composed of three people. One who is experienced in clinical trials, one epidemiologist, and one biostatistician. So all three bodies will be looking at the implementation, monitoring, safety, and everything else that happens. And this will be available, any information will be available in that that portal that I said. So this is transparency. That is why we have laid this out already. They will be there.
Ma'am, may pahabol lang ako kasi I think this is very important. Itong
Okay.
question ni
Okay.
Sheila Mantaring, no? Is
Okay.
it possible for patients who happen not to belong or fall in the chosen sites of the SVT to be allowed to volunteer or participate in the trials? I think
Okay.
that should be...
Yes. The Solidarity Vaccine Trials is under the helm of the DOST and DOH. But there are the independent ones. So the independent trialists, like I said, yung mga may bilateral agreements, I think Sputnik V, Sinovac, they have their trial zones. Actually, the trial zones will also be assigned to them by the sub-TWG on vaccine, yung DOST. But those who cannot be accommodated in the solidarity may be accommodated in those trial zones provided they meet the inclusion criteria. If they choose those sites, like if we choose barangay 1, 2, 3, we did not choose 4, 5, 6 for the solidarity.
4,
That
5,
could be
6.
chosen by the other trialists, and those they get there.
Okay.
But there will be very specific trial zones because there are specific requirements. They can volunteer,
Are
but
there benefits daw po ba for
okay.
those who volunteer?
What's the benefit? I think more monetary than anything. No, but really, if you want to participate, it may be best for the science aspect of it. But for the time you spend and the transportation, there will be a certain transportation allowance and loss of opportunity allowance. Like if you are not able to go to your work, so that will be relatively standard for each vaccinee per visit. And we envision around 3 visits for the Solidarity trial. I think for the other independent trials, they may have more. So we have standardized that. We do not want money to be the motivation here, but we want to recompense participants for loss of yung opportunity loss work, and of course travel. It's not much. I cannot say how much it is yet. It will eventually be, it is found in the informed consent form. It should be sufficient.
Thank you very much, Ma'am Nina. That was a very robust, no, our very fruitful discussions with you about our solidarity trial. So we'll be very excited to see the One Voice platform being trickled down, no? And this is a very good moment that these information on vaccines will already be transparent and open to everyone, so that no more questions or no more doubts, no? So thank you very much. That ends our open forum for you, Dr. Nina. So at this point, I'll be awarding your certificate of appreciation for being our speaker. Okay, so let me just read the citation. Philippine Network of Microbial Culture Collections Incorporated, in cooperation with Trinity University of Asia, College of Arts, Sciences and Education, Department of Biology, humbly presents this certificate of appreciation to Dr. Nina Gloriani for her invaluable contribution as our speaker for today during the 20th PNMCC Annual Symposium webinar series, 3Cs, Culture Collection and COVID-19, given on this day, 17th October 2020, signed by PNMCC President Diane Dizon and the Dean of the College of Arts, Sciences and Education, Professor Howell Ho. Congratulations, Dr. Gloriani, and thank you so much.
Thank you very much also for this opportunity.
Turning over to Caleb for our next part of the program. Thank you.
Thank you.
Thank you very much, Dr. Siddharth Siridhar. Thank you very much, Dr. Nina Gloriani. And of course, we would like to thank our webinar moderator, Dr. Marilyn Parungaw Balolong, by giving her this certificate of appreciation. You can show the certificate.
Thank you.
Maraming salamat po. Thank you very much, Dr. Marilyn Parungaw Balolong. Today's webinar would not have been possible without the help of our partners, specifically for this particular session, Diamed. For you to know more about the company, please watch this. Thank you very much, Diamed. We will now be presenting a certificate of appreciation to our partner, Diamed, as shown through their General Manager, Mr. Jose Alden Arellano, and Product and Marketing Specialist, Ms. Myla Alcantara. Maraming salamat po, Mr. Jose Alden Arellano and Ms. Myla Alcantara, representing our partner, Diamed.
Good morning, everyone. Thank you for choosing Diamed as major sponsor for today's webinar. So in behalf of our company, we would like to congratulate the organizers, PNMCC and Trinity University of Asia for this successful 20th annual symposium. Thank you, pa.
Thank you very much, Ms. Alcantara. Again, thank you, Diamed. Dr. Mike Daniel Tai of the St. Luke's College of Nursing will now give a synthesis of the two talks that we listened to a while ago. Dr. Tai?
Okay, thank you, sir. So could everyone see me right now? Okay, so actually this talk or this seminar was really a very relevant experience for all of us, especially for those who are teaching, okay, about microbiology, and also for those who are also in clinical practice, okay? Just like me, who is also a part of both the academe as well as the clinical part of this COVID-19 crisis. So we had very relevant information that were given to us, just like the review of the virus of COVID-19, which is the coronavirus, its structure, and then on how we are going to go about in the formulation of the different vaccines using the different types. Okay, it also we also had a background also on how the different protocols are being done with regards to the formulation and the testing of the COVID-19 vaccine. So what actually is the one that struck to me is there is a possibility that COVID-19, even though with the formulation of these vaccines, will still stay with us, okay, and will just be a seasonal disease probably, okay, but hopefully we will be able to develop a vaccine that can actually strengthen the immune system to be able to not only decrease the viral load, but also to destroy the presence of the virus in the body. Also, as a person who just passed the physician's licensure examinations this year, okay, so it would actually be a very big help to me, wherein I could see most of my colleagues who are in the hospitals who are also being infected with COVID-19 and some having severe symptoms. This is because of being immunocompromised. So hopefully, as we go about in bringing more vaccine trials here in the Philippines, we'll be able to develop vaccines that will actually help us to combat this pandemic that we're facing right now. So thank you, everyone, and God bless.
Thank you very much, Dr. Mike Daniel Tai. To formally close the third session of our 20th annual symposium, here is the Public Relations Officer of PNMCC and a fungal curator from the Philippine National Collection of Microorganisms, Professor Nick Sean Tabao.
Thank you, Caleb. So today we have learned so many things from the many aspects of vaccine science and technology, especially the development of vaccines in response to the current pandemic and how to choose intelligently among the different vaccine platforms to be made available or accessible to us. And of course, the role of the culture collections in all of this. We all know that COVID-19 has affected most of us in unprecedented ways. And so the talk about vaccines this morning gives us a glimmer of hope, and despite the situation, that not all is lost. But to be realistic, all of this will take time. So at this point, we would like to thank our speakers for sharing their expertise, their valuable time, and for giving us a lively discussion this morning. Personally, I would like to thank Dr. Sridhar for saying yes in a heartbeat when I invited him to give a lecture for PNMCC's webinar series, particularly today. I was following his updates in Twitter regarding the development and vaccines as early as March this year, when the COVID-19 had already reached many countries around the globe. And saying yes to a total stranger just reflects that members of the scientific community are ready to lend a hand to the rest of society when necessary. So if you would like to continue receiving updates regarding the vaccine development across the globe, I recommend following Dr. Sridhar on Twitter. So I will be sharing his Twitter handle in our FB page after the webinar. We would also like to profusely thank Dr. Gloriani for sharing her time with us despite her busy schedule. I hope that many of us will be inspired to follow the path she has carved for us Filipinos, particularly in the health and allied fields. Please do check the Solidarity Vaccine Trial updates through the Philippine Information Agency to avoid misinformation. So this event would have been impossible were it not for our partners at the Trinity University of Asia, College of Arts, Sciences, and Education, and the Department of Biology, represented by the Dean of CASE, Professor Howell Ho. So to all of you, thank you so much. The PNMCC would also like to thank our lineup of sponsors. We have ESCO, Diomed, Yana Commodities, and Lifeline. To all of you, our deepest gratitude. This webinar series is part of the PNMCC's commitment and contribution to communicating vaccine and culture collection science to the larger community. Hence, we thank you all very much for joining us today. May we all be part of the solution. So we hope to have you in the last leg of the webinar series next Saturday. That's on October 24. And for now, have a great weekend, stay safe, and may God bless us all.
Thank you very much, Professor Tabao. Before we end, please note that the last webinar will be held next Saturday, and it will focus on the establishment of Virology Science and Technology Institute of the Philippines. And then the evaluation link will appear when you quit Facebook. It can be found in the PNMCC Facebook page. It will be there for 24 hours. Certificates will be awarded to members 1 week after the last webinar, which is next Saturday, October 24. Thank you for joining today, everyone. May you have a happy Saturday.